So hello to everyone here, and welcome. Thank you to those of you who are joining us online for this event. I'm Professor Sasha Hooker, and I chair the Biology's Equality, Diversity and Inclusion Committee. I have a feedback issue going on here. Thank you, Frank. So the Biology, Equality, Diversity and Inclusion Committee, its acronym is the BEDI, or the BDI as I like to call us. I think of us as the eye on our department, looking at all aspects of equality, diversity and inclusion, and working to improve transparency and fairness in our school, across the university and beyond. As one of our initiatives, we wanted to shine a spotlight on aspirational role models. So in 2017, we began celebrating this, the UN International Day of Women and Girls in Science, by holding this open lecture to showcase the work of an eminent female biologist. All of our previous events are available online if you'd like to rewatch or watch any of these previous speakers. Today is our fifth such event, and I think it particularly fitting that we're showcasing our own Professor Anne McGurran here today. Sally Mapstone will give Anne a full introduction in just a moment. With any of these dual events, we hope to fully include those of you who are joining us online. We're planning to have a short question and answer session at the end of Anne's talk. So for those online, please do post any questions you have, and we'll try and pick these up in addition to any questions from our live audience here. Professor Frank Gunn-Moore, the head of biology, will moderate this question and answer session. We're also keen to receive any feedback on this event, so please look for the online feedback form when that arrives, and we have a paper feedback form for those of you in the audience here today. Now I want to hand over to the principal of the university, Professor Sally Mapstone, to say a few words about this event.
Good afternoon, everybody. As Sasha has just said, each year the School of Biology's EDI Committee organises a lecture to mark the United Nations International Day for Women and Girls in Science. This series provides us with an opportunity to showcase examples of the most outstanding science being done by women. And it is a real privilege in that context to welcome today's speaker, Professor Anne McGurran, CBE, FRSE, from within our own School of Biology, who will shortly deliver a talk entitled Why Biodiversity Matters. I will introduce Professor McGurran properly in just a moment, but I would first like to speak about this day and its importance to us as a university, as well as to the higher education sector more broadly. The United Nations General Assembly declared in 2015 that the 11th of February would be International Day for Women and Girls in Science, in recognition of the fact that, and I'm quoting the UN here, a significant gender gap has persisted throughout the years at all levels of science, technology, engineering and mathematics, and that even though women have made tremendous progress towards increasing their participation in higher education, they are still underrepresented in these fields. In last year's introduction for the same lecture, delivered then by the inimitable Professor Cassie Stoddard, I focused on where underrepresentation has persisted. Today, I want to take a moment to recognise how very far we have come, both because the School of Biology has a momentous accolade to celebrate, and because today's speaker attests to truly inspirational female excellence. The accolade to which I am referring is, of course, the School of Biology's attainment of a Gold Award in Advance HE's Athena Swan Charter Programme in May 2021. Each school at the university is accredited by Athena Swan at either bronze, silver or gold level, but biology is the only school to boast this highest award, and obtaining it is no small feat. It is an accomplishment of great proportions and one that few institutions can claim. It is all the more impressive because the accrediting panel commented on the level of ambition in some of the targets set in the submission. The Athena Swan Charter programme was founded in 2005 by the UK Equality Challenge Unit, which has now been subsumed into Advance HE, and the Chief Executive of Advance HE, Alison Johns, is now a non-executive member of our university court. The programme was conceived with the broad ambition of transforming gender equality within the academy, and it has delivered on that for almost 2 decades by promoting a self-assessment framework which demands a rigorous assessment of existing cultures and practices and an identification of interventions that can be made to improve them. One of the turning points for Athena Swan was in 2011, when the UK's then Chief Medical Officer, Dame Sally Davies, an honorary graduate of our university, made the ability to attain funding from the National Institution of Health Research contingent upon the applying institution holding a silver level award. At that time, there were just 10 medical institutions with Athena Swan Awards. Now there are over 145. And I remember how momentous the occasion of the Sally Davies letter felt. I was at Oxford at the time in the leadership team, and it was such a game-changing move that I had Dame Sally's letter framed, and I had it on my office wall. Since then, Athena Swan has been extended to non-science subjects and replicated around the world. In 2020, the requirement of an award as a prerequisite for NIHR funding was reversed, but the Charter programme continues to lead best practice in the sector, and it is really important for applications for NIHR funding to be very cognizant of the equality and diversity context. Now, in this general context, it should come as no surprise that the School of Biology is our first and only school to receive this award so far, because they have pioneered equality, diversity, and inclusion practices at our university. I want to acknowledge the people who have made this award possible by generating over time a school culture that genuinely prizes diversity and enables students and staff alike to flourish despite their identity. One of these people you've just heard from, Professor Sasha Hooker, an EDI leader recognized across our community as a beacon of knowledge and skill on EDI matters, and as someone with the generosity and wisdom needed to advance them. Sasha has made it clear that the environment needed to create such change in biology has come from the top of the school, in which context we should recognise its immediate former heads, Professor Gary Taylor, Professor Malcolm White, and Professor Claire Peddy, alongside the current head of school, Professor Frank Gunn-Moore, from whom we will hear later, as you know. I also wish to recognise in particular Dr. Rona Ramsey, who founded the school's EDI committee and served as its director from 2013 to 2016, laying the foundations for its current success. In truly St Andrews fashion, I know that the school has ambitions to build upon its current progress in the field of gender equality, but also further to advance its diversity initiatives aimed at supporting LGBT+, disabled and ethnic minority staff and students. Their work in this latter area is particularly distinctive already, and Professor Kevin Leyland's work in championing minority ethnic concerns and leading a decolonisation of the curriculum project in biology has set a standard to which our other schools aspire. Role modelling and standard setting, these are crucial ways that we create change by showing others what's possible. Professor Anne McGurran, today's guest speaker, is an exemplar of the very highest levels of attainment in the natural sciences, irrespective of gender. But the breadth of Anne's activities, the quality of her contributions, and the trajectory of her career serve rightly as an astonishing source of inspiration to young women scientists, both at St Andrews and more broadly. Anne received her academic training at the University of Ulster in Northern Ireland, where she was awarded the degree of Bachelor of Science in 1976 and Doctor of Philosophy in 1979. Her doctoral work focused on the biological diversity of forests in County Derry, and this informed her subsequent writing on measuring biodiversity, for which Anne is globally renowned. Anne moved from Ulster to Bangor University in 1979, where she served as a Natural Environment Research Council postdoctoral research assistant, then as a postdoctoral fellow from 1983 to 1986, followed by a further 3 years as a Royal Society University Research Fellow until 1989, and then left Bangor for the University of Oxford, where she was a Royal Society Fellow from 1989 to 1994. We finally tempted Anne to St Andrews in 1994, and she has remained here for the almost 3 decades since. Anne joined as a reader in Animal Ecology and Royal Society University Research Fellow, and she was promoted to Professor of Ecology and Evolution in 1998, a position she has held ever since. Anne served as director of the University Centre for Biological Diversity from 2013 to 2019. And she's occupied other senior leadership positions, including serving as a member of the school's management group and research committee, and more broadly serving on both the university's science strategy group and its research excellence board. Alongside these responsibilities, Anne serves as chair of the screening committee for the St Andrews Prize for the Environment, our flagship sustainability and environmental project. As a trustee and as the lead for the prize at St Andrews, I've worked with Anne in this capacity for several years, and I can thus say with both experience and sincerity that Anne has the utmost confidence of everyone involved in the prize. At the prize, as in life, Anne is renowned for the timeliness and singularity of her contributions, her ability to conceive and think around both practical and theoretical subjects, and her magnetically authoritative knowledge and understanding of the natural world. I consider myself fortunate to have worked alongside Anne, and I know others feel very similarly. Those scientists who are not fortunate enough to have known Anne personally will no doubt know of her work. Anne's first book, Ecological Diversity and Its Measurement, was published by Princeton University Press in 1988, and it is one of those sea change texts that continues to be indispensable almost 35 years after its publication, and which is available in several languages. Anne has edited 2 further books, Evolution of Biological Diversity with the Right Honourable the Lord Mayor of Oxford, published by Oxford University Press in 1999, and Biological Diversity, Frontiers in Measurement and Assessment with Brian McGill, published in 2011, also by Oxford University Press. Anne has authored an additional 2 books, Measuring Biological Diversity, published in 2004, and Evolutionary Ecology, The Guppy, published in 2005. Each of her landmark publications has been recognised as an event, and her work has been cited over 60,000 times. Whilst creating methodologies, Anne also applies them. From 2010 to 2016, Anne held a European Research Council grant for her BioTime project, in which she examined shifts in global biological diversity by quantifying changes from circa 400 ecosystems. Research as part of BioTime took place in countries ranging from Trinidad to Malaysia, and it is informing conservation events from the Amazon's floodplains forests to Europe's aquatic communities. In 2019, Anne was awarded over £300,000 of funding from the Leverhulme Trust for her project Darwin, Wallace, Bates and Biodiversity Change in the Anthropocene, which further supports BioTime in developing a framework of biodiversity change that identifies how the world's ecosystems are being reconfigured during the Anthropocene. Anne's work is of colossal environmental importance, and her authority is recognised via leadership roles in the scientific community. She has served on advisory boards at the universities of Oslo and Stellenbosch, and as a member of the ERC Consolidator Grants panel. From 2012 to 2017, she received a Royal Society Wolfson Research Merit Award, a prize designed to incentivise researchers to remain in the UK, symbolising the extraordinary demand for her expertise worldwide. And in 2012, she received an honorary doctorate from the University of Bergen, which subsequently arranged a conference in her honour. Anne has contributed internationally to research dissemination as a member of the Faculty of 1000 and has served on the Board of Reviewing Editors of Science and the editorial board of BMC Biology. She has contributed to UK biodiversity as a member of the Royal Statistical Society's Working Group on Biodiversity since 2007, and through her position on the UK Biodiversity Science Committee from 2012 to 2015. Her accolades include the Zoological Society of London Science Medal and the Beatherton Medal from the Fisheries Society of the British Isles, and she is a Fellow of the Royal Society of Edinburgh and a member of the Royal Irish Academy. I'm delighted also to acknowledge that Anne was appointed Commander of the Order of the British Empire in January 2002 for services to biodiversity. Sorry, 2022 for services to biodiversity. Through all of this, Anne has led by example as a woman in science by ensuring that the majority of her PhD students are female, with each of her former students assuming a position in the sector and actively shaping the future of the discipline. Anne's legacy is in them, in the extraordinary insight of her prolific writings and in the impact they and she have had on ecosystems around the world. So now please join me to welcome Professor Anne McGurran, CBE, FRSE, to deliver her lecture, Why Biodiversity Matters. Anne.
Thank you very much, Sally, for that very generous introduction. So the title of my talk today is Why Biodiversity Matters. It's hard to open a newspaper or listen to a news bulletin without some mention of the impact that our species is having excuse me, on our planet. And pictures from space remind us exactly how thin the skin of life on our planet actually is. This thin skin of life is what we call biodiversity, and as it's very clear that we're actually having a serious impact on the biodiversity around us. What is biodiversity? It's essentially the variety and abundance of the organisms with which we share our planet. This biodiversity provides us food, it provides us medicines, it helps regulate the climate, it provides resilience to the ecosystems around us, and as long as we keep it intact and secure, then it really guards the health of the whole planet. Once biodiversity is disrupted, then things begin to change. For example, one of the causes of the pandemics, including the one that we've just experienced, is the disruption of native ecosystems. So it is clearly to our advantage to protect this life support system of our planet. Now, we might think that concern about the environment is a very recent phenomenon, but in fact it goes back to at least the 16th century. And this is an excerpt from a lament which was originally written in Irish, and it recognised the extensive deforestation of the country that was happening at the time. So the poet here says, "What shall we do for timber? The last of the woods is down. No sound of duck or geese there, hawks cry or eagles call. No humming of the bees there, that brought honey and wax for all." So the poet here is lamenting the loss of the forest and recognising the importance that this type of habitat has in sustaining life as we know it. A more recent, a slightly more recent and rather more local example of noticing how changes in the environment are detrimental to us comes from Mary Somerville. Mary Somerville, who's on our banknotes, grew up in Burnt Island in Fife on the Firth of Forth in the late 1700s. She was a tremendous person. She was a self-educated polymath who was very well educated in physics, very influential in circles in Europe in the time, and she's the person after whom Somerville College in Oxford is named. In her autobiography, she talks about, as a child, looking out to the Firth of Forth and seeing enormous shoals of herrings coming up the Firth. The very sea was rippled by them, and whales were seen spouting in various directions. If we look out the Firth of Forth today, we won't see that sight. There are a few herring, the populations are much depleted. There are occasional whales and other cetaceans, but again, their populations are much depleted. There have been big changes in these environments, even within relatively recent history. Now, it's pretty obvious that the growth of population on the Earth in the last 100 or so years has really accelerated the extent of that impact. And as an example, I pulled out this bit of data from a recent paper, and it quantifies the amount of humans and livestock biomass. That's the total weight of everything added together. That now represents 96% of all mammal biomass on Earth. So almost everything that we see around us in terms of weight of life, and of mammal life, is accounted for by humans and our livestock. And if we think about the amount of stuff that we produce, there are more plastics by weight on the planet than there are animals. And there's more infrastructure in terms of buildings and so on than there are trees and shrubs. So we're truly having a big impact on the world around us. Now, COP26, which happened in Glasgow at the end of last year, was the first time the biodiversity crisis and the climate crisis were formally interwoven. And there has been a lot of emphasis on nature-based solutions to the climate crisis. But one of the issues about bringing about nature-based solutions, which will inevitably involve biodiversity, is that there are still substantial knowledge gaps in what we know about biodiversity. For example, in 1988, Robert May, whom Salim mentioned in the introduction, wrote a paper which was published in Science, and it asked the question, how many species are there on Earth? At that point, there were just guesstimates of how many there were. And even today, we don't know exactly how many species there are on Earth. A paper published just a couple of weeks ago in Proceedings of the National Academy of Sciences in the United States said that there are still 9,000 species of trees on the Earth yet to be formally recorded and identified. So there are substantial gaps in our knowledge. That paper also mentioned the fact that these trees are most likely to occur in places such as South America. Still a lot of work for the scientists to do in terms of recording and quantifying the amount of biodiversity we have, and then going on to understand how its roles, how the role of this biodiversity plays out in supporting the ecosystems around us. Now, to illustrate these challenges, I'm going to take you briefly to the Amazon, to the flooded forest. It's a very beautiful place, and I've been very lucky to have been able to work there over a number of years. This is a habitat in which the waters rise during the high water season by about 12 meters. They completely inundate the forests around them. And as you can imagine, the health of the rivers and the fish that inhabit them are very important in supporting local welfare and livelihoods. My colleagues, this is at the Mimiroa Sustainable Development Reserve in the Brazilian Amazon, have a large project to try and quantify the diversity of species of fish in this system. And you can get an indication here of just the amount of effort that that requires. You can see this sort of grassy vegetation. That's actually a floating meadow, and it's very important habitat for the fish that occur in these sites, particularly when the juveniles are being produced. And you can see here the research assistants are having to pull the vegetation onto the boats, sort through, identify the fish, count them, and then eventually they will return them to the water. So it's a very labor-intensive job. Now, I just got one or two graphs in this talk, and this is one example here. So if we go to any ecosystem, but particularly the tropical ones, we find a very characteristic pattern. We find one or two species that are very abundant, and many species that are very rare. So on this graph here, these are species that are ranked from very abundant ones to very rare, uncommon ones. There's a handful of species here that are very, very common, and a lot of things here, a lot of species here, which we pick up only in small numbers of individuals. This is the same plot, but I've just shown it as a blue line, just to give you a better feeling for the shape of this relationship. So in this big investigation, at that point in time, we discovered 137 species and 13,000 individual fish, so quite a big survey. Now, what we can do to try and understand this pattern a little bit better is just to stretch that vertical axis by putting on what's called a logarithmic transformation. And all it does really is to sort of pull out the abundances of the species at the bottom end of the distribution. So we have a slightly better indication of how many species in these rare classes of fish that there are. Now, this is important because we need to have that information if we're going to try and work out how many species are in the system. And this next graph just plots the same data in a slightly different format. And here we're looking at the numbers of species we detect in relation to the number of individuals we see. And the main thing to take home from this particular plot is that the red curve rises, but it doesn't flatten off. So we know that there are at least 137 species in the system, but we don't know how many there are in total. Even after sampling these 13,000 individuals, we still don't know how many species are in this system. Now, you might say, okay, that's the Amazon. We know the Amazon is an incredibly diverse system. Surely these problems don't apply in other places. Well, they do. So let's come home to Fife. And these are similar sorts of plots for two different taxa, and plotted in a similar sort of way, number of species against the number of map grid squares that have been included in the sampling. For butterflies at the top, you can see the blue line here flattens off quite nicely. So we know pretty certainly that there are between 20 and 30 butterfly species in Fife. But look at the beetles below. This red line is still shooting up. There are at least 800 beetle species, but how many more are there? So even in Fife, even with this important group of insects, we're still in the dark about how many species that we have. Now I'm going to turn now to a little section on how we might begin to understand and quantify some of these patterns. I think it's fair to say that our earliest ancestors had a very good appreciation of the fact that the species around them occur in different abundances. You can just here, I think, see there are different individuals of deer here, and one or two cows and horses and so on. So they knew that the species varied in how abundant they are. They also were quite conscious of the fact that there were they varied in how useful they are to us as humans. But it wasn't until the late 1800s and mid 1800s that people began to sort of systematically quantify this pattern. And the first one, as far as I can tell, was Darwin. And Darwin lived at this place in Down in the south of England. That was his house there. And this is a meadow beside it called Great Pucklands Meadow. And in 1855, he simply went out there and identified all the species that were there and wrote them down. This is the first quantitative survey of biodiversity that we know about. Coming into the 20th century, there were a number of mathematical advances to help us describe the patterns that we see, with Motomura, Fisher, and Preston describing patterns describing statistical models that allow us to capture the features of these plots, which are a little bit like the one I showed you from the Amazon. So we began to have a statistical representation of the data we see. And bottom right here is Alan Turing. He, as you will know, was interested in cryptography during the Second World War, and he used the frequency of letters in the alphabet to begin to deduce the codes of the enemy signals that they were trying to decipher. And you can see here immediately that letters such as E are much more frequent than letters such as Z. So it's a sort of analogous problem. So although it wasn't explicitly related to biodiversity, it was connected to it. So these were some of the first innovations in the field. As you can see immediately, it's very male. Women are not involved in this at all. And that's an excuse for me to introduce you to Rita Colwell. Rita Colwell is an extremely distinguished American scientist. She works on microbiology. She was the first woman to chair the National Science Foundation in the United States. She worked with President Clinton. She was awarded the President's Medal by George Bush. And amongst her other accolades, she's an honorary graduate of this university. So I was very privileged to serve on a science advisory board in Oslo with Rita as chair. And she made the very strong statement that when you have big problems, you need all the input, all the intelligence, and all the insights you can get. You can't exclude half the world's population from these challenges. It's absolutely essential that women play a key role in addressing these big challenges. Now, I want to mention just as well her book, which came out a year or so ago. And in it, Rita describes some of the challenges that she faced. And she mentioned that at an early part of her stage of her career, she was turned down for a fellowship because the person appointing her said, it'd be wasted on women. Why appoint her? So how wrong he was. Anyway, Rita tells us that we need to involve everybody, including women, of course, in this big challenge of addressing patterns in biodiversity and understanding how and why ecosystems change over time. Now, I've got 4 heroes of biodiversity science who are women I'd like to introduce you to. This is Chris Pilu, who was born in England in 1924, and after a number of early challenges in her career, eventually got a faculty job in Canada. She was very interested in statistical ecology, so she substantially advanced some of the other earlier methods that I mentioned. And this book, which she published in 1976, was absolutely key in my own PhD research. It really told me what I needed to do to measure biodiversity. So she's my first hero. My second hero is Georgina Mace. Georgina Mace is British. She held very, very senior positions in a number of English universities, including Imperial College. And her contribution to biodiversity science, which was really, really substantial, was to set out objective criteria for identifying species which need particular conservation attention. So she helped design the criteria that we use in what's called the IUCN Red List of Species. So whenever you see a red list of species mentioned, you will know that Georgina was part of a major part of the initiative in getting that going. She had many, many other contributions as well. Now, Georgina very sadly died just under 2 years ago from breast cancer, well before her time. But while she was alive, she made just so many important contributions to the field. My next 2 heroes are fortunately still alive. My first one is Sandra Diaz, who's an Argentinian scientist. And Sandra works mostly on plant biodiversity, but her main claim to fame at the moment is that she is the scientific lead on IPBES, which is a partner organization of IPCC. And IPBES is really international intergovernmental panel, which is concerned with biodiversity and ecosystem services. So Sandra has, in that context, been absolutely the forefront of putting biodiversity and accurate quantification of biodiversity into the policy arena. And she has won many accolades as a result. And this is just one I've mentioned here. And she's one of the people that we work with, so we're very privileged to have these connections with these great minds. My fourth hero is Anne Chow. Anne Chow is a Taiwanese statistical ecologist, and she has moved the early statistical methods on by leaps and bounds. So they are really effective in terms of helping us measure these systems, which are quite difficult to grasp and quantify. This particular picture is one that I took as a screenshot from a YouTube video, which was released this time last year for International Science Day, by Robert Caldwell. And Robert Caldwell is one of her and our collaborators. And it's a lovely tribute to her contributions. So if you're interested, you can certainly find it just by doing a quick search on YouTube. And again, Anne is somebody that we work with. It's been a real honor to work with her. And some of us are actually engaged in developing some new approaches in a project that she's actually leading. An example of that is, so I told you originally or earlier about the patterns of biodiversity in the flooded forest. And one of Anne's methods allows us to extrapolate the values that we find. And so we can work out how many species we would see if we were able to double the amount of sampling. So instead of having to go out and actually catch another 13,000 fish, we can estimate with reasonable, well, actually not reasonable, very good precision, how many species we are likely to detect. And so her methods will tell us that in that particular system, we're going to find at least 150 species of fish. And we can put confidence limits around that so we know with quite a lot of accuracy how many species are there. And you can see immediately that makes it that's a tool that really aids us in the field when we set out to measure biodiversity. Now, I thought I'd tell you a little bit about myself. So as Sally said, I grew up in Northern Ireland, and I investigated for my PhD the biodiversity of a couple of forests in County Derry. The interest of these was a combination of a relic Irish oakwood and an adjacent conifer plantation. And this was a time, I should say, when hardly anybody was interested in biodiversity, so it was interesting how things change. And so I discovered quite early on that, again, in the system, which is not a super diverse part of the world, there are major challenges in just getting down and quantifying the diversity we find about us. And so I ended up focusing on two parts of the system: the vegetation that grows in the ground and the moths that inhabit these sites. This was an interesting time because it was the height of the Troubles in Northern Ireland, and this site of Banagher Glen had been the site of a standoff between the IRA and the British Army. But I think they obviously knew I was there, but I think they both concluded that it was very eccentric, but quite harmless. So I was left alone to do my research. And these are some of the light traps that I used. And I think this is my last graph. This is using the Anne Chow method after the event to go back and compare the results I got from my two different types of woodland, my oak woodland at the top and my conifer plantation. And so even if we extrapolate the values for the conifer plantation, we find there are far fewer species of moth found there. And this is, although it was done a while ago, this is relevant to current concerns, because one of the solutions that are being posited for climate change is just to plant lots of trees. But if we plant lots of exotic conifers, then we're going to lose this possible biodiversity as a consequence. So bearing in mind the fact that we have different assemblages, different amounts of diversity associated with different types of forest, could play or should play an important role in making decisions about how we use nature to address the climate change issues that are so prominent at the moment. And as Sally mentioned as well, these initial investigations of mine were the foundation for much of the rest of my career, and they led to a number of different publications. And it's hugely gratifying to me that these books and the papers associated with them have been used by people all around the world to measure biodiversity. So that was such a nice outcome from that early work when, of course, I was trying to find my way through knowing what to do in terms of understanding the biodiversity of that little forest in Northern Ireland. Now let's turn to St Andrews. So how have women fared in terms of biodiversity research in St Andrews? Well, this is a quote which came from one of the special collections outputs, and we know that women in the early part of the history of St Andrews didn't exactly have a great deal. This particular quote says that they weren't allowed at all except if they were reasonably old, and then if they only did the laundry. But I think that reflects at that time that women had actually a very difficult challenge if they wanted to have any learning or education. During most of the history of our university, the only scope for women to have any type of learning was informal learning. So many women, for example, would know the herbs that could be used for food or for medicine. They would be aware of the poisonous plants. They would exchange this information with other women. There would be the sort of informal learning that today we applaud. We call it traditional biodiversity knowledge as a good thing. But back in those days, that could be looked on with suspicion. And if you had a cat, then that was also deemed to be a very suspicious activity. In fact, today people will use the term mad cat woman as a criticism of women. So anyway, it turns out that in 1597, James VI, who was James VI of Scotland, actually came to this university to preach, to check that preaching was up to scratch, and to attend the trials of witches. And if you know anything about the history of this area, you will know that women who had this traditional ecological knowledge were prime suspects for witchcraft. And here's just one I've pulled out of an excellent database which Edinburgh University has put together. And it's the case of a woman called Isabel D'Arcy, who was executed in St Andrews in 1643. Her crimes were witches' meetings, folk culture, diseases, and illnesses treating. So the history of women in St Andrews being interested in biodiversity type research is a little bit bleak. Now things improved dramatically towards the end of the 1800s, and we rightly celebrate advances such as Agnes Forbes' Blackadder's matriculation in 1892, a real milestone for the university. But there were still potholes on the way. In 1913, the Gatti laboratory suffered an arson attack, and it was allegedly due to an attack by suffragettes who left some notes at the scene, and one of them was, "Take heed of the women's rebellion." And although the local suffragette movement denied responsibility, there's certainly evidence that there was some discomfort amongst local women in the fact that the Gatti laboratory at that time was very much a male stronghold. And this is from the Getty History, which was written a few years ago, and it also mentioned the fact that the then director, who was called Mackintosh, was perceived to be indifferent to women, and this could possibly have been a reason for that. Things took quite a long time to change. So when I was putting this talk together, I got in contact with Peter Slater, who was the head of school when I came here first. And Peter said that he was appointed in 1984. And when he arrived, he said there were no women in the academic staff in zoology. And he mentioned Ruth Ingram in botany and Susie Whiten in anatomy. Many of us will still know Susie. As the only others in academic posts in what later became the big school. The big school, he means the combined biology and medicine, which was a school at that time and which he led. He also goes on to say that there were a number of women who were employed as demonstrators or assistants as various types. And it's actually quite hard to find any information about them. They clearly played an important role in the functioning of the university and the school, but they were very much sidelined in terms of their recognition. The big change, according to Peter Slater, was Principal Strother Arnott. And Peter describes him as a wonderful modernizer, wonderful modernizing influence. And Peter told me that when Strother Arnott came, he insisted or wanted to know whether or not there was a woman on any appointments committee, who's on the short list for any appointment. He wanted to know if there were no women, why that was the case, and he personally attended every academic interview, and he was at mine when I came here in 1994. Since then, of course, things are much better, and I think the award of the Athena Swan Gold Award is real testament for how rapidly and how importantly things have changed since then. Now, for the rest of the talk, I'm just going to tell you a little bit about some of the current research that we do that involves biodiversity. And I'll start by introducing the BioTime project. As Sally mentioned, that was part of an ERC Advanced grant, but it also involved a lot of other people, and I particularly want to mention Maria Dornelis, who has been very influential in making a lot of this work happen. So what BioTime is, it's a database of biodiversity data from around the world. And it includes what we call ecological time series. That means that you go to an assemblage, such as my Amazonian fish, and you measure it in the same way over a number of years. And then you can use those data to understand how those assemblages have changed over time. Now, BioTime is the world's largest time series of this type. It's been used extensively by researchers elsewhere, as well as by ourselves, and it has been very influential in helping us understand how biodiversity is changing over time. So I believe it's a real asset to the university. You will see from looking at this that these squares and circles show where we have data from, that they're predominantly concentrated in North America and in Europe, and also some in Australia. South America, Africa, a large part of Asia have very little data from them, and that reflects the unevenness of effort in science in the world today. Now, one of the things analysing these data have shown us is how rapidly ecosystems are changing through time. We know from first principles in science that ecosystems are not static. They're not like museum collections where you go and see the same species and the same abundances year after year. There's always some changing in terms of the relative abundance of species. But what the BioTime data is telling us is that these rearrangements of species identities and species abundances are occurring much more rapidly than we would expect, and more rapidly than ecological theory tells us. And one of the analyses that we have done has shown that this change is much more rapid in the oceans than on land. So there's very continual and intense reorganization of these natural systems, but we don't really yet understand how that is coming about or what the consequences are in terms of securing the resilience and the productivity of the systems around us. One example of the way these changes are manifested can be seen from the seas to the west coast of Scotland. We have data from there for 30 years, and when we analyse that, we find that the fish assemblages in the southern latitudes to the west of Scotland, essentially the north of Northern Ireland, and those in the northerly latitudes up around Shetland are now much more similar in species composition than they were 3 decades ago. So there's what we're calling biotic homogenization, much more similarity, much less distinctiveness in these assemblages, at least over that time period. 30 years, not very long. And a way of thinking about this is in terms of what we call the shopping mall effect, whereby if you go to a shopping mall around the world, you increasingly see the same brands in different places. There's a loss of distinctiveness in these ecosystems at least. Now that work is being followed up by Faye Moyes, who's here for her PhD work, and she's interested in how the details of how the diversity of fish in these systems is changing around Scotland, and she's already found that the changes in the west coast are different from those that are occurring in the North Sea to the east coast. Maria, I mentioned in the context of the Biotime database, but it's also important to point out that she does lots of other great stuff as well, and she works on the biodiversity of coral reefs, particularly the Great Barrier Reef. She's interested in macroecology, which is plotting these large-scale global patterns in diversity that I've shown. I mentioned that there's a scarcity of data from tropical regions, particularly this biodiversity change data. And now I'm going to tell you about some of the work that has been going on, where we've tried to gradually fill some of these gaps. Here I'm taking you to Trinidad, and I particularly want to mention Dawn Phillip. Dawn Phillip was a PhD student here at the end of the 1990s, and she went on to be an academic at the University of the West Indies in Trinidad, and we continued to work with her over the years in terms of quantifying fish biodiversity. Dawn was very influential in sharing her knowledge with students from that region, and so it's sort of nice to think that you have academic grandchildren in these parts of the world which have arisen because of the inspiration of people like Dawn. Dawn died very unexpectedly in 2017. She's still quite young, and so she is a great loss. But she's somebody that our university needs to be very proud of, I think. This work is being followed up in different ways. This is Amy Deacon. Amy was a PhD graduate here as well and worked as a postdoc with me on the larger Biotime project. And now she is also a faculty member at the university in the West Indies. And these are some of our colleagues that we were working with. This is Indar Ramlarine, who's been part of this work as well. He's currently deputy principal at the University of the West Indies in Trinidad. And Karen and Raj are field assistants who's incredibly expert about these systems. They play a really crucial part in the whole work. And Amy, of course. And current PhD students are continuing the good work. This is Ada Eslava, and she will be going off to Trinidad as soon as COVID restrictions allow, and doing her field research on fish in these waters. And Ada's particular interest is how the diversity of traits of these particular species are changing over time. The other person in the picture is Hayley Arnold, and this is Hayley doing her fieldwork, which is also in Trinidad. So Hayley looked at cocoa plantations, which have been abandoned for different lengths of time. So you can sort of see how the diversity of these systems changes in terms of the length of time over which they've been abandoned. And she measures the diversity of birds, trees, ground plants, and the epiphytes that grow in trees over time. And what Hayley has shown is that if you consume chocolate from artisanal production lines, such as those provided by these sites in Trinidad, you can actually support biodiversity. So eat chocolate, support biodiversity. Seems like a good idea. Another two of our former PhD students are Amir Ahmed and Saloni Kakon-Pisatak, and they worked on biodiversity in Malaysia, that was Amir, and in Thailand, which is Saloni. And again, they're providing really crucially needed data for these to fill these gaps in the data that we have. I also wanted to mention Anuradha. Anu did her PhD in India on the biodiversity of fish in the Western Ghats, which is a biodiversity hotspot in India. And she came here as a postdoc and worked with us in developing ways of quantifying those patterns. And finally, I still work in the Amazon with my colleagues there, particularly Elder Queiroz, who again is a PhD student from the University of St Andrews. He went on to be director of the Mamirawa Sustainable Development Institute, this area that protects the flooded forest. And this is one of the other animals that inhabit that habitat. So it's there's a wide range of mostly fishy projects, but we have other really inspirational former students and colleagues. This is Alessandra Kortz, who did her work in Brazil on what's called the Cerrado ecosystem. This is the Brazilian savanna, and it's likely to be the repository of lots of those tree species that have not yet been identified. Alessandra was interested in invasive pine trees and how they're restructuring the Cerrado ecosystem. And if you don't know, you will know now that invasive species are thought to be something that really reduces native biodiversity in many systems around the world. I also want to introduce you to Amelia Penny, who is a postdoc researcher working with Maria and myself. Amelia is integrating her understanding of biodiversity change in both fossil and contemporary ecosystems. So far, I've just talked about changes that have happened in a relatively recent past, but work by Amelia and people like her is helping us getting a much longer perspective over how these systems are changing. Amelia did her PhD in Edinburgh, and one of the things that she discovered then was a new species of pterosaur on the island of Skye. Pterosaurs are flying lizards that co-occurred with dinosaurs. So that's quite a spectacular find. At the university as well, we have Althea Davies, who's in geography and sustainable development, and Althea is interested on using pollen cores to reconstruct past environments in Scotland. And by pulling these cores out of the ground, you can identify the pollen, and you can see how the identities and abundances of different plants will have changed over time, and you can reconstruct ancient environments in that way. Alison Johnson works in Cream, and she is, again, investigating biodiversity patterns. In Alison's case, she's using citizen science data to understand how bird diversity is changing over space and time. So by citizen science data, I mean birdwatchers, ornithologists, who go out and record the numbers and identities of bird species that they've seen in particular places. By pulling all of those data together, you can have a very useful oversight of biodiversity in space and time. And Alison is at the forefront of developing new statistical methods to help us make the best use possible of those data and to get a better handle on how biodiversity is changing. We also have colleagues elsewhere. I just pulled out this example, Ilamara Smith and Gorgana Daskalova, who in Edinburgh, and they are quantifying biodiversity change in the Arctic, particularly in these shrub ecosystems. The Arctic is one of the places which is undergoing most rapid climate change at the moment. And what their work does is to bring together really state-of-the-art statistical methods with very careful on-the-ground measurement of biodiversity to quantify these changes and to allow us to manage them as effectively as possible and ameliorate them as possible. And this is my last example. This is Lorraine Hawkins, again, one of our PhD students. Lorraine is currently the director of the Salmon Fishery Trust on the River Dee, and she was highlighted in the Guardian newspaper last week for her Million Trees campaign, and the idea here is to plant 1 million trees along the River Dee to protect the fish and also to protect the habitat in that area. So the university has got a lot to be proud of in terms of all of these excellent graduates and colleagues that it has been able to be involved with in terms of doing biodiversity research. So that's mostly what I wanted to say. Still a huge amount to understand about biodiversity change, but everything that we can do to document and understand the patterns is important and will really help shape policy and reactions and protection of the natural world in a longer timescale. Before I finish completely, I just thought I would mention my aunt, Vida McCrory. Slightly different tack, but before I finish, I just wanted to make the point that we have great women doing great things. The university is a huge exemplar of that. But again, these opportunities are quite recent. We have to be very conscious of how fragile these opportunities and abilities have been. So my aunt, she was not a scientist, didn't go to university, had no opportunity, but she worked in the trade unions, and she was an active trade unionist. She was a feminist, and she worked for the civil service in London. And before she died, she told me the thing that she was most proud of in life was to be part of the campaign for equal pay for women. So before the Equal Pay Act in 1970, academic jobs, for example, I pulled one here out of New Scientist, advertised separate lower pay scales for women. That's before all the unconscious bias comes into it. So these advances in our ability and our extent to which we're treated as equal players in the system is quite recent. Needless to say, that this is work which has involved an awful lot of people from many parts of the world, and it's been a huge honour and privilege for me to work with so many great people, and a few of them are mentioned here. There are others I haven't been able to include in this slide or in this talk. But I wanted to really say that although I've focused on women, many of the men I've worked with have also been very influential in shaping our understanding of biodiversity and biodiversity change. And finally, my last slide. So I'm married to an astronomer, and this is a plate from our kitchen. And my joke is always, as the ecologist says to the astronomer, other planets cannot be as beautiful as this one. And I think that is a true observation. But what it tells me as well is that to protect this planet, to protect the biodiversity on which we depend, we need everybody to be engaged in it. So we need women and men, we need people from all around the world, we need young and old, we need citizen scientists and academic scientists to be involved. It's a big challenge, and it's something that not a single group of people can address. That's it. Thank you.
As ever in these sorts of events, I mean, it's just incredible when you sort of hear these sorts of talks. I mean, they are not just, I think, inspirational to the women in the audience, but they're inspirational to all of us, actually, about just the body of work people have done. As a humble biochemist, I always like, when I hear these sorts of talks, I sometimes think, maybe I did the wrong sort of biology. Because there is that fact that, you know, this is, everybody can see it's world-leading. Simple as that. You can see it, you know, the quality of the work. What I loved as well was all of the people you've inspired. You know, all of these people have now set up their own careers and they've kept all going. I mean, I think that truly is an exceptional thing to achieve in oneself. Really.
But the thing is, Frank, I learned as much from them as they learned from me. So.
Now, I have the task here to try and sort of multitask, because I'm going to be asking anybody here for some questions, and then I'm also going to be doing the questions from the chat as well. So I guess whilst I'll ask people here first about a question. First of all, I guess I'm going to ask you and the sort of question of, what advice would you give someone who is starting out now? Some of the younger people, whether or not they be men or women.
I think my advice would be something that genuinely interests you. So I said when I started out to work in biodiversity, I did that because I was genuinely interested in the subject. And I think if you're interested in something, you will invest more effort and passion in it. If you're interested in something, there's a good chance that other people will be interested as well. So even if it's not a popular subject at the moment, it might well become so. So follow your heart.
Absolutely. Right, so if for people who are online, if you would like to actually put your questions in the chat, I will try and pick these up at the same time. Do we have a question from the audience, preferably from somebody who isn't as grey-haired as me? Okay, we have a question here at the front.
Thank you. Thank you very much. I am not a natural scientist. I do not understand a lot of what you do, but I know it's important. I do social sciences. I'm a graduate from this university in... Okay, I'm a graduate from this university in the Legal and Constitutional Studies program, and I am currently studying a UN Masters in International Human Rights Law. As you mentioned, biodiversity is very, very important for our own benefit. We do have a lot of monocultures when it comes to places like Latin America, where I'm from, where that just happens to be a very easy, efficient way to create revenue. Of course, we would like to see more sustainable sources of agriculture, but that does imply an initial dip in revenue for when you are starting to introduce this species again. And a lot of people from these areas might not necessarily be able to make this kind of initial investment because we depend on this immediate revenue for survival. And I'm just wondering how and if scientists such as yourself or people in the field, if we're working together with the social sciences to make this more sustainable method of farming, if that makes sense.
Yes, that's a big challenge. I think the answer is that anything that can be done is good. I'm going to mention Edward Baxter, who's a farmer, landowner who lives not far from here, and he has innovated and had an innovation whereby he kept the margins of his fields much more natural and wild. And he grows cereals, big monocultures of cereals, but he's been managed by these small changes to actually support the local biodiversity. So small things like that can happen. For local farmers and so on, anything really is an improvement. Keeping hedges or small copses of trees, not extirpating the local wildlife. Those things all will make a difference. So one of the big challenges, you're absolutely right, is that many people cannot afford to do things differently. But there are these small innovations which can make a difference. And the St Andrews Prize for the Environment has actually been quite inspirational in highlighting examples of cases where people in different parts of the world have been able to do that. We had a lovely example of a case looking at working with villages who live close to gorillas in Africa, and how you could have integrated crops, health, and so on. So there are some nice examples of ways it can be done. There is some hope, I think, and having better understanding of how these small changes can make a difference is, I think, the way to go forward.
Just for people who are actually online, if you look at you want the 2 speech bubbles, which is the Q&A sort of aspect at the moment. Are there any more questions in the audience here? Don't be afraid. We have another one here.
Thank you. Thank you for the speech. I am actually come from a computer science field, and I'm looking forward to develop my career in this field, but I'm really concerned about biodiversity. So could you please shed some light on how can I relate my interest area to biodiversity? Is there anything I can do to make contribution in anything? Thank you very much.
That's a difficult one to answer.
That is a difficult one.
I think we can all do things. Some of us have more options to do things than others. So I think even within the university, there is transition, which is doing lots of practical things in terms of rewilding parts of the habitat. So there will be. Depending on your particular circumstances, there will surely be things that can be done. I think the most important thing is the willingness to get involved and to try and support whatever initiatives are there, and to start new things, and even small conversations we have about decisions people make, trivial things like what you plant in your garden. You can plant some things that support bees, for example, that are much more supportive of the whole biodiversity of the area than other things which are bred just to have pretty flowers but have no interest to pollinators. So there are any number of these small things, and I think it's great in the university that there are groups of people who get together and discuss what they can practically do to make things better.
I think I would also add on that. So if you're in the sort of computer side of things, is that what you said you were on the? Yeah. So one of the big issues now, I think we have, certainly in the whole of biology, is we have a lot of data, an enormous, sometimes too much data, and we actually need real help in actually how to sort of manage all of that data and sort of analyze that data. And certainly in the field you're in, I would say that is one of the things we really need help. Well, I certainly do anyway.
Yeah, that's absolutely true. The handling of the data is an issue, and it's going to become much more prominent once these automatic data collection approaches start up. Like, I don't know if you've heard of environmental DNA, where people go out and take samples and sequence it and see what's there. There are other issues involved in that, but one of them is going to be data handling. So that's definitely. And there's certainly lots of projects you can do even here if you're interested in biodiversity themed things.
Do we have any more questions in the audience? I think there's maybe 1 or 2 issues on the sort of chat. I'm not seeing any sort of questions at the moment, but there's a question at the back there. I can see. I think it's Inika.
Hi, Anne. Thank you so much for your talk. As you know, I'm a mathematician, so I loved your log graphs. And I was struck by the graph that highlighted the difference in biodiversity between the oak forest you were looking at and the conifers. And as you know, because of offsetting, you know, we've had these conifer forests shooting up everywhere, and they're quick to grow, whereas an oak forest takes a very long time to mature. How do we balance that tension of perhaps needing these forests to go up quickly because of the kind of idea of offsetting CO2 and supporting biodiversity with these much slower native forests?
I think we have to, we're in this for the long term. I think we have to do things which will endure for long periods of time. So it's perfectly possible to have a balanced solution that has some short-term benefits, but also takes into account the longer-term benefits as well. I don't think it's either or. I mean, I think it's something we need to think through and think carefully about, and not immediately do something because it seems to be the right thing, but rather to weigh up the advantages and disadvantages of different strategies.
Have more question here at the front. You have a microphone just coming to you so that people can then hear the recording.
That was inspiring. Thank you. In your travels in indigenous areas, have you found that the wisdom of the indigenous people, tribal people, and their knowledge about over thousands of years, you know, sustaining their natural environment, have they been able to find a market through fair trade? I'm very interested in fair trade and forest honey and forest cocoa or coffee, all that sort of thing, which aims to not only provide a decent of living, but to conserve the natural environment. It's
okay. Yes. Yeah, so probably two answers to that. First of all, to flag up, it's not enterprise for the environment again. That has got some lovely examples of supporting people in producing outputs and trade and so on that actually benefits the local communities. But if I can talk about the Mamirawa Reserve, which is this big reserve in the Amazon, sort of half the size of Wales, the people there have been working with the local inhabitants to initiate sustainable solutions that help them. And so one thing that they've done is ecotourism. Another thing that they have done is to have exports of ornamental fish, which people will know are sustainably development. They can put sustainably harvested. They can put a little tag in them. So if you have this fish with some kind of cichlid in your fish tank, you can screen its microchip like putting a cat's back, and you can see exactly where it comes from. You know, its provenance is very ethically sourced. So they have that sort of thing, but also. The Mamirawa Reserve has got an educational program for the people who live there, and it is providing training for them post-secondary school so they can get professional qualifications if they want. If they want to go into science, that is an opportunity for them as well. So yes, so there's certainly examples that we know of where these things are absolutely core values. In terms of the field assistants, I mean, I showed you a number of them, and they're incredibly expert. They know what the different species of fish are by looking at them. They won't necessarily know what their scientific name is or where they sit in the evolutionary tree, but it is an interplay between their knowledge and the scientific knowledge. And in terms of Mamirawa, when that reserve was established, which was back in 1990 by a very inspirational biologist called Marcio Ares, the people who were living there had quite a low standard of living, very high child mortality, a lot of disease, very low income. The quality of their life has just rocketed. They're just so much better off than where they were at the start of the project. And every time I go back, I can see the well-being, the quality of life is just so much better. So yes, these things are compatible, and they're examples where it can be made to work.
Do we have another question? I'll even take a question from somebody who's not is grayer than me. not casting any comment on his hair.
Thanks. I'm just curious that any of the data you're collecting on microorganisms?
We haven't studied microorganisms, and that's a big unknown. As the DNA technology comes much more to the fore, then there are increasingly large studies of microorganisms, and that's so two things are relevant there. One is that Rita Caldwell was a microbiologist, so she was a sort of leader in that field in identifying the diversity of taxa. But also the methods I told you about by Anne Chow are increasingly used by microbiologists. And one bit of work that I didn't talk about, where we had started characterizing the species that occur in some of these ecosystems, we discovered that there are some species which are there most of the time, we call it core species, and there's some species which are transient species, they come in and out. And I received a very interesting email a few years ago from somebody who was working on microbiomes, and they said, but we find exactly the same pattern there. So you can use the inferences we get from studying our fish communities to understand microbial communities, including our microbiome. And I think as the microbiome research advances, that there will be increasingly synergy between that work and the sort of thing I've talked about here.
Do we have one more question from the audience here? If not, then it just remains me to do a small thing. So Anne, if you don't mind, I'm just going to pop behind you. Yeah, excuse me. Obviously, thank you so much for such a wonderful talk and everything, and in a small appreciation from the School of Biology, we'd like to give you this gift. Okay, thank you very much. Thank you for such a wonderful talk, Anne, and thanks for being such an inspiration. Thank you.